Welcome, students. Today, we are going to understand one of the most fundamental concepts in biology, the genetic code. This is the language of life that connects DNA to proteins. By the end of this lesson, you will clearly understand how information stored in genes is translated into functional proteins. The genetic code is a set of rules that defines how the nucleotide sequence of DNA, or mRNA, is translated into the amino acid sequence of a protein. In simple terms, codons determine which amino acid is added at each position in a protein.
The genetic code is extremely important because it connects genetic information with protein synthesis. Proteins are responsible for the formation and functioning of enzymes, hormones, receptors, and many cellular structures.
Now let us move to the history of the genetic code. George Gamow proposed that genetic information could be read in the form of nucleotide combinations. His idea provided an important theoretical foundation for understanding the triplet nature of the code. A group of 3 nucleotides is called a codon. With 4 different bases arranged in 3 positions, a total of 64 possible codons can be formed. The genetic code was experimentally deciphered through the enormous contributions of Marshall Nirenberg and Har Gobind Khorana. Using different synthetic RNA sequences, researchers determined which codons specify which amino acids. Severo Ochoa's enzyme, polynucleotide phosphorylase, helped scientists synthesize RNA-like polynucleotides in the laboratory. These synthetic RNA molecules became valuable tools for deciphering the genetic code experimentally. In Nirenberg's famous experiment, synthetic poly-RNA was used in a cell-free protein synthesis system using Severo Ochoa enzyme, that is, polynucleotide phosphorylase. This RNA produced a polypeptide containing only phenylalanine. This clearly established that the codon UUU, which is a homopolymer, specifies phenylalanine.
Har Gobind Khorana synthesized chemically defined repeating nucleotide sequences to decode the remaining codons of the genetic code. These experiments systematically established the relationship between codons and amino acids.
Let's study the salient features of genetic code. The genetic code is triplet. Each codon consists of 3 nucleotides. 4 different bases form a total of 64 possible codons. The genetic code is degenerate, meaning more than one codon can specify the same amino acid. For example, UCU, UCC, UCA, UCG all code for serine. However, the code is unambiguous. Each codon specifies only one amino acid. For example, UUU always code for phenylalanine.
AUG has dual functions. It acts as the start codon, or initiator codon, and also codes for methionine.
The 3 stop codons, UAA, UAG, and UGA, terminate translation. The code is nearly universal. For example, from bacteria to human. UUU would code for phenylalanine, Phe. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. The genetic code is non-overlapping. Nucleotides in one codon are not shared with the next codon. The code is commaless and continuous. There are no punctuation marks between codons. Let's quickly revise how the genetic code was deciphered. George Gamow proposed the triplet nature of the genetic code. Severo Ochoa provided the enzyme polynucleotide phosphorylase, which helped scientists synthesize artificial RNA. These synthetic RNAs included homopolymers, RNA made of only one type of nucleotide, and copolymers, containing different combinations of nucleotides. Nirenberg and Matthaei used a poly-U homopolymer and made the first breakthrough, showing that UUU codes for phenylalanine. Har Gobind Khorana then used chemically defined RNA sequences, including copolymers, to decipher additional codons. Together, these discoveries led to the complete genetic code, 61 sense codons for 20 amino acids and 3 stop codons, UAA, UAG, and UGA. Remember the sequence: Gamow, Ochoa, Homopolymers and Copolymers, Nirenberg and Matthaei, Khorana, complete genetic code. It is now time to practice the previous year questions. These questions will help you revise the concepts and identify the topics that are repeatedly tested in examinations. Keep exploring the wonders of molecular biology, and I'll see you in the next lesson. Thank you for watching.